Welcome to all of you. Myself, Dr. Rahul Shankar, Faculty, Science and Technology, Vajiram and Ravi. In the journey of preparation of civil services examination within the general studies paper, we have the GS paper 3 for Science and Technology section. This Science and Technology section is also linked with the general science portion. If you look into the syllabus of prelims, then general science is one of the important components of the syllabus. And the other component is science and technology for the mains exam. In fact, the chapters of science and technology, they are also asked in the prelims exam. Now, in case of science and technology, particularly the current affairs portion, that plays a very significant role for this exam. Here in the institute, we are going to have the discussion on a wider platform, not only confined to the topic itself, but also in terms of the domain of the knowledge that will be required for the interview also, including prelims, mains, and the interview. In fact, as it is linked with the current affairs, so newspaper analysis is also a very integral part of it. And the past 10 to 11 months before the prelims and before the mains exam, that form the basis for the asking question for the examiner. So including some of the Nobel prizes also linked with the science and technology section, that also because they reflect the importance of certain technology innovation for the mankind over the years and how we are going to move forward and how it can be applied at grassroots level. Now, for example, in terms of science and technology, so for example, a topic on general science, if you look into the Nipah virus disease. So this is a topic that is based on current affairs, how, and that is in the past 10 to 11 months, this was in news and the questions could be asked from this area. How do we discuss this topic so that you have the brief overview and also related to the wider aspect of the topic. Nipah virus disease, Sungai Nipah. Sungai Nipah, this is the name of the village in Malaysia, where the first outbreak was reported in the late 1990s. 1998-99. Followed by this Nipah virus disease that also got spread to the other Southeast Asian nations, including Singapore, Cambodia, Philippines, then Bangladesh, and later on in 2001, for the first time from Bangladesh, it also entered India from Siliguri in West Bengal. Nipah virus disease was found. For the first time in Deep South, it was in 2018 May that in Deep South in Kerala, the disease was reported. Now, Nipah virus disease, it comes under zoonotic disease, where so Niv here means Nipah virus. So this is a bat-borne virus. Bat-borne virus. And these belong to the Pteropodidae family of bats, which are frugivorous, nectivorous bats. So bats are the primary reservoir host. Primary reservoir host of this virus. Primary reservoir host means virus in the body of bats, they have adapted together. The virus adapted to live in the body of the bats, and bats they have natural antibodies. They have natural antibodies against the virus, due to which bats do not fall sick in the presence of the virus. Virus may multiply in their body, whether faster or slower, depending upon their immunity of the bats. However, the presence of bats will not affect the presence of virus in the body of bats will not affect the bats. So they do not develop signs and symptoms of the virus. Now, for the Nipah virus, intermediate host are the pigs. Intermediate host are the pigs. And the explanation regarding transmission of Nipah virus, one important concept known as zoonotic spillover is very important. So zoonotic spillover means accidental transfer of virus from animals to the human or bats to the human. How it occurs? The zoonotic spillover. Deforestation. Deforestation increases human, animal, environment interaction. So this means this will increase the contact of human with the bats because of deforestation, and this will increase the risk of mutation, and zoonotic spillover. Increasing the risk of transmission of Nipah virus as a bat-borne virus. Now, in case of bats to human transmission, what are the different modes of transmission of the Nipah virus? So here when we discuss in the class, then you'll find that we are also going to have the notes that you need regarding the revision in terms of the comprehensive notes, apart from the classroom explanation, from where the questions may not be able to, that is spillover, that is in terms of the whatever questions are going to come, you will find that they will revolve around the classroom discussion. So Nipah virus, zoonotic virus, meaning it can spread in animals and people, and pigs are the intermediate and amplifying host. So pigs are intermediate and amplifying host. One of the important infection symptom, that is encephalitis, viral encephalitis, where there could be neurological symptoms. So when we are going to discuss in the class, then we'll have the broader in terms of understanding how do we connect with the classroom discussion in terms of not only the written part, but also, as we can see here, that how fruit bats with Nipah virus, if they feed on the date palm sap. So this date palm sap, this is the pteropus bats, the fruit bat that we discussed. So their urine saliva can contaminate the date palm sap or juice that people are collecting. Then consumption of this raw sap can infect the human. At the same time, human to human transmission, any infected body fluid, it could be sweat, saliva, respiratory droplets, any of the body fluids can transmit the virus. Then at the same time, there could be bats to pig transmission. So contaminated fruit with bat bite, with the saliva of bat, can infect the pigs. And these infected pigs, when they come into contact with animal husbandry workers who may not be using gloves, mask, or while slaughtering the pigs, handling the pigs, there could be transmission of infection. Then pig meat, pork, that consumption also can transmit the virus if it is not properly cooked. Then export of pork to other countries also can carry the virus. So that is in terms of the broader transmission of the Nipah virus, how it occurs exactly. So consideration should be given while establishing pig farming in the areas where there is a natural presence of the pteropus bats. In terms of signs and symptoms, if you look into, then we discuss involvement of respiratory illness, the lungs here, involvement of brain also, encephalitis, and encephalitis that results in the symptoms of drowsiness, disorientation, mental confusion, These are the typical symptoms. It can also result in symptoms of seizure and even coma and death. And here, the most important part, case fatality rate is very high. Case fatality rate means out of 1,000 infected individual, what proportion of risk of death is associated. So out of 1,000 infected individual, there is a risk of 750 to 1,050 to 900 deaths. So there could be 75% to 90% case fatality rate in Nipah virus disease. So that is important, that case fatality rate (75% to 90%), which is quite high. So this is why there is a serious concern on the outbreak of Nipah virus, that if at all it converts into a pandemic as a zoonotic outbreak, then it can play havoc for the society. So we have to take measures to contain the outbreak of the Nipah virus disease. So this is how an important topic that we discuss in the class related to general science. Then related to the current affairs also, how we have to connect the topic. It keeps on changing every year and we have to update ourselves. For example, Nobel Prize Chemistry of 2023. Now this was regarding the quantum dots. It was regarding what? Synthesis of quantum dots. Now these quantum dots, these are semiconductor nanocrystals of less than 10 nanometer size. For example, cadmium, selenide. Now the quantum dot, this is also the extension of discussion of quantum mechanics. In quantum mechanics, the behavior of subatomic particles, electrons, photons, that is peculiar at the nanoscale. So subatomic particles, that is electrons, for example, subatomic particle. So they show the property of duality. That is, they will act as a wave also and as a particle. If we confine the movement of electrons at the nanoscale, less than 10 nanometer, then it is acting as a wave and they will show size dependent optical properties. They will show out size dependent optical properties. So quantum dots, when they confine the movement of electron as a wave, so depending upon the size, quantum dot size, they also show their optical properties, which means greater size or larger quantum dots, they emit color of, so large size quantum dots, they emit color of longer wavelength, or so we know that frequency is inversely proportional to wavelength. If they emit longer wavelength or the red color, so this means their frequency is lower. So they emit color of lower frequency and longer wavelength. Similarly, if the size of quantum dot decreases, then they are going to emit higher frequency color and shorter wavelength, that is the blue color. So quantum dots synthesis can produce different sizes or different colors. And this different color quantum dots, this can be used in QLED television, quantum LED television. It can also be used for biological imaging of tumors, or in terms of biochemistry also, imaging of various biomolecules, it can be used as a practical application. So now we are looking into that is how the scientists, they have introduced the quantum dots. So we can see here the Nobel Prize in Chemistry 2023 was awarded to Mongi, Bevendi, Bruce, Alexei for the discovery and synthesis of quantum dots. Further, we can see now that quantum effects are when particles shrink. So when particle, we can see here larger nanoparticles. So we discussed that these are semiconductor nanocrystals. So larger nanoparticles, they have more space for electron wave. So they are going to emit color of lower frequency, longer wavelength. If we reduce the size of quantum dot, then electrons are getting confined. So smaller nanoparticle, less space for the electron wave. And that is why they will emit what? Of higher frequency or shorter wavelength, blue color. So these multiple color quantum dots can be synthesized, or this Nobel laureate, they synthesized and they proved that how this can be further applied, these particle dependent size in terms of, so when particles are just few nanometer in diameter, the space available to electron shrinks, and this affects particle optical properties. So in terms of application, so this topic is important both for prelims and mains. How do we deal with? So in science and technology, there could be a combination of some topics exclusively for prelims, exclusively for mains, or there could be a combination. So where this topic can be asked both in prelims and mains exam. So this is another example that luminous or fluorescent properties of quantum dots are utilized in computer and television screen based on QLED technology. Now Q stands for quantum dot. Then doctors have begun investigating potential quantum dot to track tumor tissue in the body. So therefore the nanotechnology also is an integral part of this, the Nobel laureates in terms of finding the properties of the quantum dots. Similarly, there is another that is important area, that is in terms of artificial intelligence. So one of the portion that we'll discuss as a science and technology is the artificial intelligence, which is defined as an algorithm or set of instructions to perform a task. AI emulates human intelligence to rise to the same level of reasoning and thinking as a human. So there are certain complex problem solving tasks which can be also achieved by using what? Machine intelligence. So AI is an algorithm. Learning algorithm is called machine learning. So machine learning is a branch of artificial intelligence, where machine learning focuses on increasing the accuracy, of predictable outcome. Focus is on accuracy, a predictable outcome, where the important task is forecasting using the machine learning. So this machine learning, including AI, so that is widely used now in diverse areas. So this area, it touches everyone's life now, whether it is the positive aspects of AI or some of the implications related to the AI. So positive aspects means that how this AI, it can be used for clinical diagnosis. And this was also the question that was asked in the mains exam of 2023. So 2022 mains exam, they asked on the, I think that is in the 2023 they asked. We'll see the question. So we will also focus in our classroom discussion regarding the mains exam also, that how to write answers and how the questions they are also updating and how we have to update according to the question. So you can see here, introducing the concept of the AI, and then how AI can help in clinical diagnosis, and do you perceive any threat? What are the concerns on AI regarding data privacy, data security, the concerns. So we'll focus in the classroom that how we have to approach and write the answer to the questions, where in limited words, that is 150 words, how we can write the answer to a question. Although it comes from experience. In civil services, the three important attributes which are required for an aspirant. One is the consistency, another one is perseverance, and the third one is the determination. So these three are the most important attributes that we must have in terms of while preparing for the civil service examination. Now, one of the key aspect of AI in clinical diagnosis is medical imaging and early disease detection. So when we go for any type of medical diagnosis, for example, where X-ray or CT scan, MRI, and that is interpreted by the radiologists, followed by the differential diagnosis by the concerned physician. Now, in this case, artificial intelligence in the form of machine learning, that also can identify changes in the pattern of the data. These changes in the pattern of the data, they can identify the hotspot of decision making. That is the valuable aspect of AI. So, for example, to examine the density of tissues, how it has been changing in terms of the breast cancer or in terms of lung cancer. So that can be well analyzed by AI also. It can pick up in terms of that which person or is showing the signs of the breast cancer or the lung cancer. Then early disease detection is very important. So we know that in cancer diagnosis or in the disease diagnosis, even in diabetes also. So early diagnosis is very important because early diagnosis brings better results, improve the quality of life, shelf life of the individual, and also better results in terms of medical outcome. So AI can be used to identify early signs of disease. So early signs of disease, it can potentiate the role of doctors in disease diagnosis, revolutionize. Now, for example, before we go to the concerns, in terms of diabetes retinopathy, one is the cancer detection, diabetes retinopathy. So in case of diabetes mellitus, there could be damage to the retina of the eyes in terms of the blood vessels that could be damaged, which can cause loss of vision. So early detection of diabetic retinopathy is very important. Otherwise, in late stage diagnosis, it can cause loss of vision. So that can be better analyzed, well predicted by the artificial intelligence. It can also be used for genome sequencing, DNA sequencing for risk assessment. It can early stages of cancer, genetic disease can be predicted. So there is immense potential of AI in the healthcare or clinical diagnosis. Even in mental health issues like Alzheimer's disease also it can be used in clinical diagnosis. Now, in terms of concerns here, we can see there are concerns on data security, that the data that they have taken, whether the medical colleges or the hospitals, they have acquired the data of patient, whether they are able to protect the data, the sensitive data. At the same time, data misuse could be there. So they have to take informed consent of the individual for transparency. So data misuse means the AI system healthcare providers could misuse patient data for purpose other than diagnosis treatment. Suppose it could be misuse patient data, that diagnosis treatments and marketing or in terms of insurance or research. So there must be avoidance of data misuse, that is protecting the data privacy. De-identification. So generally they go for data masking, so that the personal details of individual, the name, address could be masked. However, this even after demasking, that is masking of the data, there could be de-identification or re-identification of those data. So de-identification challenges that even if the data has been anonymized, the same machine learning can tomorrow be used to join the dots and re-identify the same individual. The breach of data privacy can occur. Then other issues like informed consent we discussed, algorithm bias, yes. So in AI, one of the important concern is the biasness in algorithm. That how there could be concerns regarding empathy, societal consciousness in AI. In this case, in clinical diagnosis, it could create a prejudice, stereotype for a certain group of people or population that they are only prone to certain genetic disease or certain type of ailments. So that can create significant discrimination of a population. So that also must be avoided by using the AI. So discriminating against certain demographic groups, so that can also occur. So there are challenges. So this is how the questions are in mains exam, and we look into the discussion in the class. Then one of the important breakthrough of this century, the greatest breakthrough in terms of what? mRNA vaccine. In the context of that is COVID-19 pandemic, if we look into, one of the important contribution by the scientists have been introduction of effective mRNA vaccine. Effective mRNA vaccine. Developed world, including US, UK, European Union, they relied more on what? mRNA vaccine, which was first introduced by the Pfizer company. followed by the Moderna mRNA vaccine. So questions in exam, they are asked related to what? The current affairs and how they relate to the benefit for the humanity. They try to coincide both of them. So in that context, this is the very latest development and also a very important development in the healthcare industry regarding the mRNA vaccine. So there are certain future applications. First of all, in mRNA vaccine, there were certain challenges that were addressed by the Nobel Prize Medicine 2023. So for example, earlier mRNA vaccine that used to cause increased risk of heart attack, blood clots, excessive inflammatory reactions, and lower efficacy of the vaccine. But these were addressed by the Nobel Prize Medicine 2023. What they did, they discovered that what is the component present in the structure of mRNA that is responsible for increased risk of heart attack or blood clots, inflammatory reactions. Two scientists, Kathleen Carrico and Drew Weissman, they were awarded Nobel Prize Medicine regarding the development of effective mRNA COVID-19 vaccine, and that changed the lives of millions of people worldwide, and it is also going to revolutionize the healthcare further. So how innovation can be done with equity, that is also a concern, that it must result in shared prosperity in the society. mRNA vaccine, as we discussed, the two challenges. The third challenge lies in effective delivery of mRNA vaccine. mRNA delivery, as mRNA has a very shorter shelf life, it is unstable in nature. So we need an effective delivery system. Standalone mRNA will be destroyed by the enzymes of the cell. So we need lipid nanoparticle to deliver mRNA vaccine into the cell so that it can withstand attack by the enzymes of the cell, and then it can produce the desired effect. Now regarding this Nobel Prize, we can see in case of mRNA, the presence of nucleoside base uridine. So this nucleoside base uridine that resulted in inflammatory response here. At the same time, lower efficacy of the vaccine, the spike protein. We are considering in the terms of what? COVID-19 vaccine. Any COVID-19 vaccine, it has to work against what? Production of antibodies against the spike protein of SARS-CoV-2 virus. But previously, mRNA vaccine resulted in excessive immune reaction, as you can see, and lower expression of protein. Inflammatory response. However, chemical modification, nucleoside base modification of uridine to pseudouridine, that resulted in higher efficacy of spike protein, increasing efficacy of vaccine, and also avoiding this inflammatory response. And this is how they were able to realize the effective use of mRNA vaccine in the world. Then another point is, generally any type of vaccine that we know, like inactivated, like in India we use the Covishield viral vector vaccine, we use Covaxin as the inactivated vaccine. All of these required what? Cell culture. Whereas mRNA vaccine does not need cell culture. Therefore, there could be scalability in mRNA vaccine production. In a limited time, record time, we can produce it. Because during COVID-19, when the pandemic struck the entire world, then the first concern was how to develop quickly an effective vaccine, where mRNA came as a rescue, a great opportunity in the crisis. Because without cell culture, it can be produced, it provides scalability of vaccine production. Then we can also update the vaccine quickly depending upon mutation in the virus. So that is another aspect of mRNA vaccine. India has also developed now its own first indigenous mRNA vaccine. So in exam, you may get a question, one is the concept, another one is the current affairs. Current affairs like, what is India's first mRNA vaccine? First indigenous mRNA vaccine. And it is nothing but the Gemcovac biopharmaceutical company developed. So India Genova biopharmaceutical company developed Gemcovac OM. This is the first indigenous mRNA vaccine of India. OM means it has been developed against Omicron variant of SARS-CoV-2 virus. Omicron variant of SARS-CoV-2 virus. So we can see here, this is the spike protein of the virus, spike protein of SARS-CoV-2 virus. So this is responsible for attaching to our lung cells to cause infection of that symptoms of breathlessness. Now, in order to block the virus attachment, our body should produce what? Immunity should produce specific antibody to block the virus attachment. So mRNA vaccine can help develop these anti-SARS-CoV-2 antibodies. So any vaccine help develop what? Memory cell. And memory cell, when they come into contact later on with the real virus, then they are going to produce anti-SARS-CoV-2 antibody, circulates in the blood to block the virus infection. So this anti-SARS-CoV-2 antibody, they block the virus attachment or infection and protect the body. So this is the India's first indigenous mRNA vaccine, Gemcovac-OM, and targeted against what? Omicron variant of SARS-CoV-2 virus. So we can quickly update mRNA vaccine depending upon the type or variant of virus. So we can develop variant-proof vaccine. We can develop what? Variant-proof vaccine using mRNA technology platform. Then mRNA vaccine has the future potential in terms of developing cancer vaccines, therapeutic proteins to treat various genetic diseases, and also vaccines against those diseases that we have not yet developed any, that is the specific vaccine for it. For example, MERS-CoV, HIV, AIDS, do not have any vaccine. Even Zika virus disease also. So we can develop the vaccines against certain other diseases where we do not have any vaccine. So that is the future application, potential of mRNA technology platform. Now, one concern is that definitely that is in terms of the ultra cold chain into minus 70 degrees centigrade in terms of the storage transport. But that has been addressed in the Indian Gemcovac-OM, because now it can even at 2 to 8 degrees centigrade it can be stored. That India already has this infrastructure in terms of low temperature infrastructure or logistics. So that is on the mRNA vaccine. So that is why this is how we are going to discuss in the class, that how the classroom discussion should be updated in terms of the demand of the exam, whether prelims or mains exam, how we have to approach the topic in a comprehensive manner. So that is what we are going to have in the classroom discussion. So I welcome you all. Good luck to all of you. Thank you.